Welcome to the weekly Neurology Deep Dive, where we take complex topics and distill them into essential knowledge for you, our dedicated listeners. This deep dive is produced by Ameriga Vanini. Today, we're embarking on a deep dive into a fascinating and, frankly, quite challenging disorder, Valosin-Containing Protein, or VCP myopathy. You might also know this condition as Multisystem Proteinopathy 1, or MSP1. And historically, it was called Inclusion Body Myopathy, Paget's disease, and Frontotemporal Dementia, often abbreviated as IBMPFD because of that classic triad of symptoms.
Right. And before we jump in, it's really important to clarify that this deep dive is strictly for educational purposes. There are no commercial objectives here. Our goal is just to summarize the source material provided, and, for a complete understanding, we strongly recommend that you consult the full research papers.
Absolutely. So for those of you who need the critical takeaways right now, maybe you're short on time, here's a concise overview. VCP myopathy is a complex autosomal dominant disorder. So a single copy of the mutated gene from one parent is enough to cause it. It stems from mutations in the VCP gene, and this leads to widespread protein aggregation and dysfunction in the cell's really crucial protein degradation pathways. Clinically, its presentation is remarkably variable. It frequently includes progressive muscle weakness, bone disease, dementia, and sometimes even amyotrophic lateral sclerosis, or ALS.
And diagnosing it requires a high clinical suspicion. This is then supported by electrophysiological studies, specific muscle imaging patterns, and ultimately, of course, definitive genetic testing. Recent large-scale muscle biopsy studies have consistently shown myopathic features, things like atrophic fibers, rimmed vacuoles, protein aggregates. They also reveal these unique degenerative niches, which might suggest how the disease actually spreads, and these appear regardless of the specific VCP gene variant. Current treatment is mainly symptomatic, but preclinical research into VCP inhibitors is showing some genuinely encouraging results.
That's a great summary to start us off. Now, let's explore what makes VCP myopathy such a, puzzling and important area in neurology. Let's unpack the VCP protein itself first. It's often called P97, and it's described as this highly conserved, ubiquitously expressed ATPase. What exactly is its core job in our cells?
Well, what's truly fascinating about the VCP, or P97, is its critical role in maintaining cellular proteostasis as protein homeostasis. You can sort of think of it as a central hub in the cell's recycling and quality control system. VCP belongs to the AAA plus protein family, and it uses energy from ATP hydrolysis, essentially, breaking down ATP for energy to remodel molecules. It's a key player in the ubiquitin-proteasome system, which is the cell's primary pathway for tagging and breaking down misfolded or damaged proteins. And it's also deeply involved in endoplasmic reticulum-associated degradation, clearing out proteins that go wrong during their synthesis in the ER. But beyond those critical functions, VCP also plays a significant part in autophagy. That's another crucial cellular process for degrading and recycling cellular components, including misfolded proteins and damaged organelles. And its reach extends even further. It participates in diverse functions like membrane fusion, cell cycle control, maintaining genomic integrity, and even the formation and clearance of stress granules. It's really a multitasker.
Okay, so it's involved in so many vital processes. What happens then when mutations occur in the VCP gene? How does such a crucial protein go, awry?
That really is the key question. Pathogenic mutations in VCP disrupt these processes, and that leads to the hallmark pathological feature of the disease: the accumulation of ubiquitin-positive protein aggregates in affected tissues. The precise mechanism is still being worked out, but it's believed to involve impaired protein clearance and dysfunctional mitochondrial quality control. Interestingly, many pathogenic VCP variants are associated with an increase in ATPase activity, which is intriguing, right? It suggests a gain-of-function mechanism. So the mutated protein doesn't just stop working, it actually becomes overactive. And this overactivity is thought to lead to excessive degradation of crucial proteins, like mitofusin, for example, contributing to widespread cellular damage.
It sounds like disrupting such a central regulator would naturally have broad consequences. And that leads us to the clinical presentation, which, as the sources tell us, is incredibly variable. You might expect a really clear set of symptoms, but even within the same family, it can manifest very differently. How common is that classic triad: inclusion, body myositis, Paget's disease, and frontotemporal dementia?
You're absolutely right about the variability. That classic IBM-PFD triad is actually seen in only a minority of patients, somewhere between about 3 to 12%. Far more common are these, more variable presentations.
Okay, let's break down the major components of this variability, starting with the muscle involvement, often presenting as inclusion body myopathy.
Right. This typically shows up in mid-adulthood, usually between, say, 30 and 50 years of age. patients experience progressive muscle weakness and atrophy, and it frequently starts in the pelvic and shoulder girdle muscles, the proximal muscles. This can translate into everyday difficulties like climbing stairs or raising your arms, and it eventually progresses to generalized weakness. In the later stages, it can unfortunately involve respiratory and cardiac muscles, potentially leading to life-threatening breathing difficulties and even heart failure. Muscle biopsies in these cases often show progressive vacuolization of myofibrils, centrally located nuclei, and cytoplasmic accumulation of TDP-43 and ubiquitin-positive inclusion bodies. And while rimmed vacuoles are characteristic, they can sometimes be infrequent, surprisingly.
Okay. Beyond the muscles, another significant aspect is the impact on the skeleton, specifically Paget disease of the bone. What does that look like?
Paget disease of the bone occurs in approximately 50% of individuals with VCP myopathy. The mean age of onset is around 40 years. It's characterized by, well, excessive bone breakdown and formation, leading to abnormally large and weak bones. It most commonly affects the hips, spine, skull, and long bones. And this can cause significant bone pain, skeletal deformities, and an increased susceptibility to fractures. Although, interestingly, it might even be asymptomatic in some patients.
And then we have the cognitive side, frontotemporal dementia, or FTD.
Yes, FTD affects approximately 30% of individuals with VCP myopathy, often with an earlier age of onset than typical FTD, usually becoming noticeable in a person's 40s or 50s. Symptoms involve a progressive decline in cognitive functions, particularly personality, behavior, and language. You might start subtly, maybe difficulties remembering words or names, trouble with numbers, comprehension deficits. As it progresses, patients can experience significant personality changes, a loss of judgment, inappropriate social behavior, and eventually lose the ability to speak, read, or care for themselves. Brain tissue from these patients often shows intranuclear TDP-43 positive inclusions.
And VCP mutations are also linked to ALS, aren't they? How does that connection manifest itself?
That's correct. VCP mutations have been linked to familial ALS, accounting for about 1 to 2% of those cases. This form is characterized by progressive degeneration of both upper and lower motor neurons. That leads to muscle weakness, atrophy, fasciculation spasticity, and bulbar dysfunction, which means difficulty speaking and swallowing. And importantly, both ALS and FTD and VCP disease commonly exhibit TDP-43 aggregates in the affected tissues, which really points towards a shared underlying pathology.
Given its role as this kind of master regulator in the cell, are there other associated features or perhaps surprising presentations we should be aware of beyond these main ones?
Oh, absolutely. The multisystemic nature means there are several other things to look out for. These can include distal myopathy, peripheral neuropathy, for instance. Charcot-Marie-Tooth disease type 2Y has been linked to parkinsonism, cardiomyopathy, hepatic steatosis, even sphincter disturbance. And furthermore, a pediatric form of VCP deficiency has recently been identified. This presents with developmental disorders, intellectual disability, hypotonia, and macrocephaly. So that significantly broadens the phenotypic spectrum, showing it can impact individuals right from a very young age.
So we understand it's a genetic disorder. Where exactly is the VCP gene located on the chromosomes, and what specific types of mutations are we typically seeing?
The VCP gene is located on chromosome 9p13p12. And as we mentioned, the inheritance pattern is autosomal dominant. Over 50 heterozygous missense mutations have been identified so far. They predominantly cluster in the N-terminal half of the protein, which is a really crucial region involved in ubiquitin binding and various protein interactions. The arginine 155 to histidine substitution, the R155H change, is the most common VCP mutation. It's found in over 50% of patients presenting with that classic inclusion body myositis, Paget's disease, and frontotemporal dementia triad.
Okay, so given this incredibly complex and variable presentation, how do neurologists typically navigate this landscape to arrive at a definitive diagnosis? What are the key diagnostic tools in the toolkit?
Well, diagnosing VCP myopathy really relies on putting together a puzzle using a combination of clinical features, imaging, electrophysiological studies, muscle biopsy, and of course, genetic testing. Clinical suspicion is usually raised first by the presence of proximal myopathy, especially if there's a family history of muscle weakness, bone disease, or dementia. Electromyography, the EMG, may show myopathic changes, but sometimes you also see neurogenic changes, which could indicate a coexisting peripheral neuropathy or motor neuron involvement. Muscle imaging, particularly MRI, can provide a very useful diagnostic clue. It often shows a relatively specific pattern of muscle involvement. You'll predominantly see affection of the parascapular, paraspinal, gluteal, and quadriceps muscles. And often you'll see positive S-star sequences. That's a type of MRI sequence good at showing fluid or inflammation indicating edema. But ultimately, targeted gene panel testing using next-generation sequencing is the definitive diagnostic method to identify those pathogenic VCP variants.
And speaking of diagnostics, there was a recent large international study that really dove deep into the muscle biopsy findings. Can you tell us a bit more about the scope and significance of that research?
Yes, that study by Isiava and colleagues, published in 2025, is quite significant. It involved 112 muscle biopsies from patients across 52 centers in 24 countries. So it's the largest comprehensive analysis of VCP-related myopathology to date. The study found that about 66% of individuals in their cohort were male. The mean age at biopsy was about 53 years, and the mean disease duration was around 6.5 years. The most frequent VCP variant they saw was that PR155 his change in about 19% of patients. And the most common clinical phenotypes observed in this particular biopsy cohort were isolated myopathy in about 38% of patients, then myopathy with Paget disease of the bone in about 18%, and myopathy with motor neuron involvement in about 13%.
Right. So what did those biopsies actually reveal about the disease's impact on the muscle tissue itself? What were the common threads?
Well, the biopsies provided pretty consistent insights. About 91% were open biopsies, with the vastus lateralis being the most common site, chosen in about 35% of cases. The most frequent histopathologic findings were quite widespread. They included atrophic fibers in about 88% of samples, rimmed vacuoles in about 72%, endomysial fibrosis in about 58%, and protein aggregates in about 52%. These protein aggregates primarily stain for P62, that's a protein involved in the cell's waste disposal system, in about 50% of cases, and for VCP itself in about 36%. While they weren't routinely stained for, TDP43 and ubiquitin staining were identified in some biopsies too. Inflammatory cells were detected in about 30% of the muscle samples, mostly macrophages surrounding necrotic fibers. But expression of major histocompatibility complexes class, I was restricted to only a few fibers, which is a pattern unlike what you see in other inflammatory myopathies.
That's interesting. And one of the more novel findings mentioned was these degenerative niches. What exactly are those, and what might they signify about the disease process itself?
Yes, these are truly fascinating, a really significant new observation from that study. About 31% of the biopsies showed these demarcated focal areas, which the researchers termed degenerative niches. They're characterized by fibers with structural alterations, things like internal nuclei and P62 inclusions, and also markedly atrophied fibers, all embedded in fibrotic and fatty tissue. What's particularly striking is that these foci can be located within areas of otherwise pretty normal-looking muscle tissue. Importantly, the study found no significant differences in how often these degenerative niches appeared among the different clinical phenotypes. That suggests they might be a common feature across the disease's varied presentations. Now, there were generally no differences in biopsy findings among the 4 most common VCP gene variants, but there was one notable exception. They found an absence of these degeneration niches in the muscle biopsies of 12 patients who had the PR93CC's variant. That had a p-value less than 0.01, so that suggests there might be some variant-specific pathological differences, even within the general consistency.
That's interesting. Did these muscle biopsy findings, these niches, correlate with what was seen on muscle MRI?
Yes, they did find a clear correlation. Muscle MRI data was available for 30 patients in the study. They observed fat pockets. These appear as sort of rounded areas of fat replacement inside otherwise normal muscle in about 47% of these patients. And these could indeed correspond to those degenerative foci seen on the biopsy. Additionally, about 70% of the muscle MRIs showed that increased signal on SDR sequencing, which is compatible with muscle edema. And of those with S-dye changes, about 43% also exhibited inflammatory infiltrates on biopsy, and about 33% showed the presence of macrophages. So a strong correlation between the imaging and the histological inflammatory markers.
It really sounds like this level of heterogeneity, both clinically and pathologically, must make diagnoses particularly challenging for a busy neurologist in practice.
It absolutely does, and the study underscored this. It revealed a significant challenge. Concordance, meaning agreement, between the clinical phenotype, the muscle biopsy findings, and the needle EMG findings was observed in only about 49% of cases. That starkly highlights the substantial heterogeneity in VCP multisystemic proteinopathy. And it reinforces why a high index of suspicion is really required to prevent underdiagnosis and potentially misdiagnosis. While the muscle biopsy findings are characteristic, as we said, they aren't pathognomonic, meaning they aren't unique only to this condition and can overlap with others. However, Their consistent presence is a critical diagnostic clue that should definitely prompt further investigation, including genetic testing.
Right. So, understanding the complexity of diagnosis, what does all this mean for a patient's prognosis? And what's the current state of treatment for VCP myopathy?
Well, VCP myopathy is unfortunately a progressive disorder. Affected individuals typically experience a significant decline in muscle strength over time, often leading eventually to respiratory and cardiac failure. The prognosis is variable. It depends on the specific manifestations and their severity in each individual. But typically, individuals live into their 50s or 60s. The involvement of respiratory and cardiac muscles can, as you can imagine, be life-threatening.
And is there currently a cure available for this condition?
Unfortunately, no. There is no cure for VCP myopathy at this time. Treatment primarily focuses on symptomatic management and supportive care, really aiming to improve quality of life. This includes things like physical and occupational therapy to help maintain muscle function and independence for as long as possible. Also, respiratory support for patients with respiratory muscle weakness, cardiac management for those with cardiomyopathy, pain management, especially for Paget disease of the bone, and cognitive support for individuals who develop frontotemporal dementia.
Okay, so while there's no cure yet, are there promising research avenues being explored? Are there things on the horizon that offer hope for future therapies?
Yes, absolutely. Significant research efforts are underway, and there's definitely hope here. Given that the mutant VCP protein seems to be abnormally overactive, promoting excessive degradation of essential proteins, VCP inhibitors are seen as a highly promising therapeutic strategy. For instance, a novel VCP inhibitor called CB5083 has shown encouraging results in preclinical studies. It improved motor behavior and ameliorated muscle pathology in VCP-R155H knock-in mouse models and also in patient-derived myoblasts in the lab. Other strategies are also being explored, like looking into autophagy modulators to try and restore those dysfunctional pathways we discussed earlier. And gene therapy approaches, such as antisense oligonucleotides, are being considered, although those are still in the very early stages. Importantly, mouse models and human-induced pluripotent stem cell, or iPSC-derived cell models, are proving to be invaluable tools for these preclinical studies and drug development. They allow researchers to really dissect the disease mechanisms and test potential treatments before moving to humans.
So we've taken quite a deep dive today into VCP myopathy, clearly a very complex and heterogeneous condition impacting muscle, bone, and brain. It's certainly a challenging disorder, but it does seem like one where diagnostic clarity and hopefully future therapies are steadily emerging.
Exactly. And just to reiterate our practical summary for your daily clinical practice, maintain that high index of suspicion for VCP myopathy, given its incredibly varied presentation. Remember, genetic testing is definitive for diagnosis. This is supported by characteristic, though not pathognomonic, muscle biopsy findings. Think atrophic fibers, rimmed vacuoles, protein aggregates, and those distinctive degenerative niches. Specific muscle MRI patterns can also be very helpful. While current management remains supportive, the exciting preclinical results, particularly with VCP inhibitors, offer a strong signal of hope for future targeted therapies that could potentially fundamentally alter the course of the disease.
It feels like the ongoing research into its molecular pathogenesis is truly paving the way for more effective treatments, hopefully transforming the lives of individuals affected by this really challenging disease.
And just thinking about the bigger picture, the progressive expansion of those degenerative niches in the muscle. It raises an important question for future research. Could VCP myopathy involve some kind of spreading, maybe a prion-like mechanism of protein aggregation? Similar perhaps to what we suspect in other neurodegenerative conditions, that's certainly a compelling area for continued exploration.
A fascinating thought to end on. Thank you for joining us on this weekly neurology deep dive produced by Ameriga Vanini. Remember to consult the full research papers for complete details. We look forward to our next deep dive with you.